Hello everyone, welcome back to the part 2 of GA signaling mechanism. In my previous lecture, we learned that the Gibberellin signaling requires a receptor, a soluble receptor called as the GID1 receptor, and there is a villain of the signaling called as the DELA repressor. The DELA repressor has two domains, N-terminal regulatory DELA domain and the C-terminal repressor domain called as the GRAS domain. So there was an N-terminal called as DELA and C-terminal called as GRAS. In my previous lecture, I showed you that when no Gibberellin is there, right, what is going to happen in a particular plant cell and why the signaling is not happening, right? So when no GA is there, why no signaling is happening? Because when Gibberellin was absent in a plant cell, the signaling was blocked by DELA repressor and more specifically the GRAS region, right? What region? The GRAS domain. So I'll repeat once again, when Gibberellin was not there, the GRAS domain of the repressor, right, was kind of blocking the activator, right? I hope you remember. Therefore, there was no signaling. So when Gibberellin enters into the cell, it is going to take care of this particular problem, right? So let's see what is going to happen, right? So this is the picture that when Gibberellin was not there, the GRAS region of the DELA repressor blocked activator, therefore promoter was blocked and responsive genes not working. You can have a look at my previous lecture for that. But the question here, or the part that I am about to discuss, is not that why signaling was not happening earlier, but we will try to understand what chain of events will happen when Gibberellin comes into a plant cell. So Gibberellin comes along with its receptor called as the GID1 receptor, shown in green, right? So in blue color we have shown this Gibberellin, right? This is GA, right? GA is always bound to its receptor, shown in green, called as the GID1 receptor. But always remember that these two never come alone. They always come along with a particular protein called as the GID2 protein, which is an F-box protein. Please remember, in molecular biology, F-box is a protein which can degrade any target protein. It can degrade that protein by simply attaching ubiquitin groups to a target protein, and then that protein will be finished off in a cell with the help of something called as 26S proteasome. But that is not the question here. What we are trying to understand is how GID1, GA and GID2 actually degrade or finish off the DELA repressor. Let's have a look at that. So when this combination comes in, please remember the Gibberellin or GA can recognize the DELA domain of the DELA repressor, right? So Gibberellin bound to its receptor, it goes and it removes this DELA repressor from here, right, by binding to which domain? The N-terminal DELA domain. So the role is clear. DELA domain is required for being recognized by the Gibberellin so that it can be removed by Gibberellin, while the GRAS domain was actually shutting down the signaling mechanism, right? So what we have here is now this particular combination, right? This particular combination of Gibberellin plus GID1, this removes this GRAS and DELA domain containing GRAS repressor from here, right? So as you can see, the GRAS plus the DELA domain containing DELA repressor will actually be removed from here. So this is kind of a good news for the activator. So you can see that the activator kind of becomes happy, right? So activator is now happy. Why it is happy? Because DELA repressor has been taken away by Gibberellin. Now DELA domain was identified by Gibberellin, but what is happening to the GRAS domain? The GID2 F-box protein sits over the GRAS domain, and once GID2 attaches to any protein, I told you, once GID2, which is a type of F-box, once it attaches itself to a target protein, that is the, in this case, the DELA repressor, that will be degraded or shattered into pieces via the 26S proteasome, which is a part of molecular biology information. Now, the activator is happy. So if the activator is happy, what will happen? So the activator will activate the promoter will activate the gene transcription will happen, GA signaling will be switched on. So I once again repeat, in presence of Gibberellin, the Gibberellin never comes alone, it always comes along with GID1 receptor, plus there is a following protein which is coming along with that team called as GID2. Now GA recognizes the dela region. Please remember, if something happens to the dela region of a dela repressor, Gibberellin can never identify that protein, right? So please remember, dela domain is that region which is being utilized for recognition by GA, as a result of which on the grass domain the GIT2 sits and the dela repressor is finished. If repressor is finished, activator, as you can see, is happy, promoter active, response genes active, and as a result of this, GA signaling is going to happen. So that's all from the signaling point of view. I have one question for you which is quite famous in examination, then we can finish off our topic. The question says that what will happen to a plant if there is a particular mutation in the dela domain. So I just told you that dela domain is the region which is required if Gibberellin can go to that dela repressor and remove it from the activator, right? So please understand, if you mutate somehow, if anything happens to the dela domain of a dela repressor, therefore repressor will never be degraded by GA, because Gibberellin will never be able to identify that mutated dela region, right? So if dela region is mutated, GA will keep revolving around the repressor which was sitting on the activator, and as a result of which the repressor can never be removed. So if repressor can never be removed, that it is cannot be degraded, therefore there will be no signaling happening. Signaling of GA is not possible even when GA is present. Doesn't matter, even if you spray Gibberellin from outside, it is not going to happen. As a result of which, if no GA signaling is there, such a plant is going to be a GA insensitive dwarf. Please remember, sometimes students might get confused. If in exam it is asked that mutation in dela domain is which kind of mutation, so this kind of mutation in molecular biology is a gain of function mutation with respect to the repressor. Please remember, this is a gain of function mutation with respect to the repressor. Why gain of function mutation? Because the repressor got some power that it can never be degraded by GA. Please remember, this mutation in the dela domain made or gave the dela repressor a power that it can never be finished. Now since it has gained some power, so it is called as gain of function of mutation with respect to repressor. Repressor can never be degraded if dela domain is improper, as a result of which GA signaling will always be off even when GA is there in a cell, as a result of which such a plant will always be dwarf in height. It can never increase its height no matter how much quantity of Gibberellin is provided externally. Now if this is the case where we have for mutation in dela domain, let's try to understand what is going to happen if there is a mutation in the grass domain, and then we will finish off with this chapter, right, or this topic. So what my next question is, what is going to happen if there is a mutation in the grass repressor domain, right? First of all, please try to understand. So what will happen? So if you mutate what grass repressor domain, because that domain was actually shutting down the activator. So if the grass domain is kind of mutated, therefore repressor will not be able to shut down signaling. Repressor will be inactive, you can say. Repressor will be inactive. Even in absence of GA. So I hope you understand. Even when Gibberellin is not there in a cell, the repressor is not working. It is not able to shut down the activator, right? So that means, can I say activator always active? So activator always active, right? As a result of which signaling always active. Signaling will always be active, right? So if signaling is always active, right? So even when Gibberellin is not there, what was Gibberellin doing? Gibberellin was removing the repressor, right? But now your mutation has made the repressor inactive. As a result of which signaling will keep on happening. This is called as constitutive GA signaling, right? It will keep on happening, as a result of which signaling will always going to take place, and this will be a plant with increasing height. It is going to increase its height always, right? This is called as a slender phenotype, right? So this is called as a slender phenotype. slender phenotype. I have a question for you. I hope you can now understand this function, right? Can anyone tell me, right? Please write the answer in comment if you like this video. So just like mutation in DELA domain was called as a loss of function, it was called as a gain of function, sorry. So mutation in the DELA domain was called as what? Gain of function, because the repressor was never being degraded. What will you call the mutation in GRAS domain as? Will it be a loss of function or will it be a gain of function? Please leave your reply in the comment. I will try to incorporate the correct answer as quickly as possible. And that will be all for today from my side. The Gibberellin signaling is finished. It was one of the most requested topics from the students, and part 2 is now over. If you have anything else for me, do let me know. The video would be uploaded for that topic shortly. Thank you and have a nice day.